So, Robin, here we are again.
Yeah.
We tried the first round, and now this is the official launch of our podcast, Big Ideas at the Micro Scale. And I think when we last left off, we had said that we were going to start bringing on some really interesting teams,
hmm
and I'm so happy to start off with the team out of Cambridge. This Filip and Iwan were amazing. When we first spoke to them, I think it was in 2018 or 2019. I got a chance to meet them in 2023 at their lab, and the work that they've been doing has been quite amazing. I guess before I go any further, I'd like to introduce myself. I'm Hemdeep, co-founder of CADWorks 3D, and I've got my co-host, Robin.
Hello.
How are you doing, Robin? You've been digging out of all this snow over the last, I guess, 4 days now.
Yeah, there was really a lot. It's not fun. My arms hurt. I think what was it? 30 centimeters of snow over 2 days. I think it was.
Yes, yeah. It was a barn burner. It really was a lot of snow. So, all right. So let's start with this conversation. I'm happy to introduce Iwan and Filip to our first inaugural podcast, and so here they are. Yay!
Thanks a lot for having us on. It's a great honor to be the first ones.
Yeah, thank you very much.
I'm going to rewind this to, I guess, some of the more important starting points for you guys. I guess we would want to know, exactly what your backgrounds are, what your relationship is, and sort of where the connection point between the two of you started off.
You want to start?
Yeah, why not? So yeah, really, to be honest, a lot of the connection came through 3D printing between us, really. So I started actually in January 2020 in our lab in the University of Cambridge. I started as a postdoctoral researcher, really looking at making electrical phantoms of the cochlea, which is the little structure which looks like a snail in the middle in your inner ear. And basically the first week that I started, I was tasked by our supervisor to buy a 3D printer like this week and just so figure out which kind of printer you want to get. And then I met Filip, who just started his PhD at this, just a few months earlier, and we kind of went through and tried to really kind of look at what kind of printer we'd want to get at that point, which ended up being a longer process than 1 week. So because the structure of the cochlea is so small and intricate, we were really looking at printers that could replicate that really kind of fine anatomy, and especially the small hollow channels. And that kind of snowballed over time to doing all sorts of really wonderful prints, mainly related to the ear anatomy, and eventually kind of has come into us co-founding a company about a year and a half ago called Cochlea, which specializes in 3D printing different sorts of anatomies and getting really fine structures.
And you, Filip, how do you connect in with Iwan, and what kind of specialty, skills do you bring into this team?
Yeah, so I've met Iwan first time in a couple of months after starting my PhD, where Iwan joined as a postdoc, and as he mentioned, the first task was to get a 3D printer, and it's a bit funny because now looking at it back, like getting the task to select the 3D printer for very special use case, and giving a timeline of 5 days was a bit crazy and ambitious. But before starting the PhD, which was in clinical neuroscience, I did a engineering degree back in Czech Republic, which was in nanomaterials and nanotechnology. It's like basically the equivalent of Master of Science. And then I did Master of Philosophy or MPhil here in Cambridge, which was about micro and nanotechnology in enterprise. And it should have been like 50% science, 50% business. But it ended up in good old 95% science, 5% business. But I really like the environment around Cambridge and the university itself. So I applied for the PhD and got in, worked with Manohar for a couple of months. And then Iwan joined in. We started working on selecting the 3D printer, and then we start the actual work, where particularly my research was a lot about 3D printing cochlea or the inner ear, where specifically what I was looking at is that I think that maybe I should introduce a little bit the cochlear implants, because that was the core research, and it still is part of the research in the lab. Cochlear implants are these devices which helps people with tremendous hearing loss to hear again. It's a device which can bypass normal hearing. So how it works is that if you have very high level of hearing loss, so imagine no hearing, you are a great adept for cochlear implants, and you basically need to undergo a brain surgery where they open the skin or cut the skin behind the ear, drill into temporal bone, create mastoidectomy, and then that creates an access to the inner ear, where they then can put the cochlear implant, which is basically a wire with a couple of electrodes covered in silicon. And this way you can directly stimulate the auditory nerves, which are located inside of the cochlea, the spiral shaped like object. But even though this technology is incredible, because it basically gives you a sense which you might have lost over time or you were born without, there are a couple of problems. So one of the problems is that if the patient has any residual hearing, there is a quite high probability they will lose it during the insertion. So because there are insertion forces that arise, and then you can basically mess it up as you putting in it, because it's a very small object and quite fragile as well. And that was basically my research. So I was looking at how these insertion forces arise and how we can mitigate them to preserve the residual hearing of patients. So then the patient could sort of benefit from both type of hearings, the auditory one and the electric one, which is done by...
And if I jump in, the time that I was at your lab, you guys showed me that stage that actually measures, measures the forces, the insertion forces. Now, was that did that require a completely set of engineering in order to understand how to even measure those internal forces?
Yes. So part of the work was to create this insertion setup where we use quite sensitive sensors to measure the insertion force. So it's a setup where you can load up a cochlear implant, and on one sensor and on the other sensor you can put the 3D printed cochlear model, and then you can slowly insert the implant in while you have multiple cameras, and all the recordings as it goes in, and you measure the insertion force profile, and then you can sort of look at how the geometry or the anatomy of the cochlea could make a difference on these insertion forces.
Like, how many specialties did you have to bring in order to sort of develop? It sounds like even before you got to actually testing it, there was the set of protocols that you had to develop was involved a significant number of skills to bring into place.
I think that our background is sort of scattered all over the place, considering the fact that, maybe Iwan, you can tell a little bit about your background, because it's also a little bit special.
Yeah, so my background is a little bit strange in a way. So I originally did a master's in physics, so very pure physics, going into a little bit of biophysics. Went more into the bio during my PhD in Manchester, UK, working in tissue engineering and regenerative medicine, where we were looking at engineering effectively scar tissue and muscle tissue in a dish, and looking at the mechanics of how that the cells would remodel their matrix or the materials that they sit in. And then I came kind of in this sort of interface between that with the postdoc work, kind of bridging the kind of electrical physics kind of aspects with the biomaterials kind of aspects. But this kind of is a really good question, Hemdeep, in terms of the multidisciplinary nature of our kind of work, because as well as us two, there's obviously quite more of a team as well, which look at both the mechanical aspects for this, but also more biological aspects and also clinical as well. So we have people coming from more of a clinical side, so ENT surgeons who come to do a PhD or different research projects with us, as well as people coming from more pure biology, looking at sort of a Cochlea-on-a-Chip project that we've done that we can go into a little bit on the microfluidics side, as well as some more on computational aspects, where really trying to simulate both on a physical level and a computational level, especially the electrical interactions between the cochlear implant and the nerves, because that's kind of the key component here, as well as once you make sure you have a nice insertion, which makes sure it preserves the native neural population, you want to be able to most efficiently stimulate those nerves to give you the correct sound that you want to have.
Yeah, so I was kind of curious, who actually brought both of you to cochlear implants? Like, was it one of your primary research topics, or were you building on a different research?
So I knew nothing about cochlear implants before doing the PhD. So that's also interesting, because then from that perspective, the PhD was in clinical neuroscience, but I'm very much an engineer rather than a clinician. But the way I joined is that when I was doing my MPhil here in Cambridge, I was working in the material and metallurgy department. My supervisor recommended me to Manohar, and during the MPhil, I was working with a very special type of 3D printer called or printing technology called aerosol jet printing, which is this unique type of technology. And I was working on a project to create basically these flexible, stretchable microbatteries, which could potentially power cochlear implants. And yeah, that's the first time I heard about cochlear implant. And after finishing up that project, I started talking to Professor Manohar Benz, who then became my supervisor on PhD.
Speaking of properties, I think that was part of your PhD project, right? It was focusing mainly on the material properties of like the implant arrays and how that affected the insertion forces. Is that right?
Yes, partially. Basically, the PhD itself, what I was trying to investigate is that because there is a range of anatomies related to the cochlea, so generally speaking, there are different shape and sizes of cochleas. So every cochlea is slightly different.
Scala tympani, right?
Yes. So
one of the chambers.
Yes. So the cochlea itself is divided into like these 3 chambers. And the one I was mainly interested in is the scala tympani one, where the implant actually goes. And these chambers, or the scala tympani chamber in this case, or the cochlea, could be of different sizes. And I was interested in how these these might affect the insertion forces, and if there is any sort of relation between, let's say, if the insertion force is higher, if that results in higher probability of losing the residual hearing, or if, let's say, robotic insertion could help out with reducing the trauma and hence preserving the residual hearing. So the PhD itself was about studying these forces, generally speaking, and part of that was to segment the shapes of the cochleas from micro CTs and create a workflow where we could 3D print these reliably with a certain level of deviation, and then improve the properties of the prints to be representative of the insertion forces, which can be also measured in cadavers. So there was a lot of sort of work and side projects along the way, and part of that was also creating the insertion setup.
So in terms of your data set, then how big was this data set that you needed to work off before you felt that you had a viable starting point, and the insertion forces and the data that you were starting to obtain from all of your setup was now not only theoretical, but it actually was something you can derive an application from?
Mm-hmm, mm-hmm. So I also have to add in that a lot of the work was a teamwork. So it wasn't just me working on this. There are people working on the scanning and characterization of the cochlear shapes, and we were able to get about 90 cochleas to be segmented out and fully characterized, which create the pool. And then out of the pool, we were able to check different properties and select the cochleas based on these extremes. So what I was interested in, looking at, average volume versus very big one, very small one. Then other aspects. In one of the papers we published, we look at one particular shape of the cochlea, and then we artificially change it. So we could measure the specific parameter and it change on the insertion force. That's also one of the things which we really, really like, and I think it's important to mention about 3D printing, is that it gives you ability to change just one parameter at a time and measure it multiple times. And then this way you create you create results which can be built upon. Because the problem is that if you start with complicated structure right at the start, and you get another complicated structure which has which is different in 5 different parameters, it's difficult to then measure which parameter actually plays the significant role and makes the biggest difference. So one thing we explore is that we took one average cochlea and then we artificially change one parameter at a time and measure the insertion force, 10 times, 100 times to get reliable data. And then we change another parameter again. And then this way we were able to compare the insertion forces between these different parameters and say which parameter is actually important.
I was curious, what are the main parameters that you guys identified?
So there's a fair bit of characterization to do actually on the small cochlea. So we have some kind of small complex shape like this, which is probably too small for the camera to see, where because it's an ascending spiral complex shape, it's one of the worst kind of shapes to use engineering wise, because in every direction is kind of complicated. And we've got to shout out Chloe Swords, who is a PhD student coming from a clinical background, who did a ton of work on this in terms of cutting down samples and scanning and a lot of the characterization of these cochleas, as well as 3D printing phantoms directly with them, which we collaborated closely. So there's kind of a workflow that we've developed, which we're preparing some publications on as well now, where we can take a template from a synchrotron micro CT, which is effectively a particle accelerator, which will generate really high power X-rays to conduct a very good micro CT effectively, where you have much better contrast. And what that allows you to do is to see the individual chambers of the cochlea, or the microstructure within the cochlea. And using that template, we've worked with colleagues in engineering, such as Professor Andrew G, to manipulate this template to fit other micro CT data, as well as clinical grade CT, where, because you can see the outline of the shape, you can fit this template on. And what I did afterwards is to effectively characterize from different landmarks that we have along this like spiral shape, characterize the cross-sectional area, the overall size, the volume, everything under the sun that we can think of to characterize this type of shape. Because a lot of people would say, oh, well, you want to compare what's the influence of the anatomy on different factors, but you need to know what the anatomy changes would be when you're actually doing this. Otherwise, it can be sometimes in the literature quite vague, like we've just chosen a big cochlea or a small cochlea, but maybe the curvature is quite different, and that might be the key determining factor in the forces, for example, or maybe the shape or the angle of approach. Surgical approach might be quite difficult. So there's lots of different considerations to think about. And now we've generated this large data set of about 90 cochleas that Filip mentioned. We can then see what the population would look like and where that kind of average cochlea would be, and see what the extremes then would consist of it. And one other thing that we found in the paper we recently published was that for, at least on the insertion forces, when we made a mathematical model of how the insertion forces should change due to the anatomy, it really all boiled down to how many degrees you've went around the spiral, where it effectively comes in this capstan model, which is actually, funnily enough, used for like very old engineering principle of how many times you tie a rope around a bollard on the side of a dock, the force exponentially increases. But rather than going around the kind of one bollard, you go on the inside of a structure. You can use the same principles mathematically. It's kind of equivalent. So once we changed the size and shape, actually, you accommodate for the angular insertion depth around this little structure, then the force is all aligned, even on a bigger cochlea or a smaller cochlea.
So now, as you're doing this, the lab work, how frequently are you then reconnecting with clinicians or those that are on the surgical side in order to validate what it is that you're doing, or validate at least, are they are surgeons looking at you and saying, okay, if we, these are the steps or the angle at which we do our approach, and then how does that impact the type of research that you're doing on your side then?
Yeah, so I think this is really key, really, is the validation of the work. And actually, I think one thing I should mention is Professor Manohar Bans, who leads the academic lab, is a practicing ENT surgeon. So he's been an ENT surgeon for almost 30 years now. So has a wealth of experience, and so guides a lot of the research questions that we would try and engineer. So he gives us problems and we try to engineer solutions effectively. So it is pretty integral to the lab. And for example, Chloe, who I mentioned, it comes from an ENT background. So she really brought that insight as well. And it's really interesting to see the different perspectives you get in terms of the practical kind of, oh, how do I use this in my practice effectively, versus, oh, how do you simplify that into really understanding on a mechanistic level what the key influences are. So, for example, like we've got the very detailed work looking at the forces on the cochlea itself, but then we also need to look at how do you secure the lead during the after you've put the cochlear implant in, you need to make sure the lead is secured. Otherwise, that might actually lead to the cochlear implant coming out and actually destroy all the good work that you've done. So we've worked with Dr. Clark and Dr. Tom Hudson on quite a lot of work on that, and seeing how you might do different drilling techniques or different ways that you might secure the lead to make sure that it's secure over long-term use, and actually that you're not generating more trauma after you've been very careful putting it in and just like, okay, let me tie things up at the end, and actually making the thing worse.
So now when you start developing all of that, was there an actual aha moment, a moment where you're just like, I think we're sort of you're sort of there's traction in what you're doing, and there's the feedback that you're getting from clinicians seem to indicate that you are sort of moving in a fairly positive direction?
Yes, like in terms of the research itself, the advantage there, like we were discussing this with the ENT surgeons here at Edinburgh's hospital, which is the hospital at the biomedical campus of the university. Or it's a university hospital, and at the I think one of the, it wasn't more like, it wasn't an aha moment, rather than like a disbelief that the model could actually follow up, let's say, the Capstan equation, and that that fairly simplistic solution could actually work out for the insertion forces, like to describe the insertion forces in cochlea. So we spend a lot of time to prove that works, and there was a lot of back and forward regarding publishing the paper as well, because there was this belief that the simple equation could describe the insertion forces. I think that the aha moment or the real surprise was when we 3D printed the models and treated them in the way that the insertion forces started to look similar to the one in cadavers. Because we knew from literature and from experience what the insertion forces should be. And once we find the solution to treat the inside of the lumen or inside of the channel in a way that the insertion felt and behaved as during the cadaveric work, then we thought that we are on something. And then once we start looking at, because we were worried that this treatment could actually change the accuracy of these models, so we spent a lot of time to create a technique to double check that the models are still fine and they are within a certain deviation. Once we got all this work done, then we were sort of sure that we have one of the best models to describe the cochlea and to do this type of work.
If you're telling me that the development of the internal model was the actual aha moment, can you describe what that was like? What steps you needed to do in order to start developing an accurate internal cochlear model? Like, what was the work that you guys had to do before you got to that point?
First of all, you really need to start with a good micro CT scan. We quickly realized that CT scan is simply not good enough to create a great segmentation. Just so you can sort of understand what I'm trying to say is that cochlea is so small, is that if you take a normal hospital CT scan, you will have about 7 scans where the cochlea actually is shown. So 7 slices. Whereas if you use micro CT, it's about 700. So then you can really pick up small features and you can create a high resolution model. And that's very important for any 3D printing, I would say.
And what was the scale? Sorry, what was the scale at which some of the features were at in terms of millimeter or micron? What sizes were you working with?
Please, Iwan, jump in if I say something incorrectly. But the entrance, so let's say the round window, which is the entry point for the cochlear implants, is about 1 to 1.5 millimeters. And then as you follow up the channel and it starts to spiral up a little bit, it becomes smaller and smaller. By the end, we're talking about 300 micrometers, I would say. 200, 300, depends. It's sort of changed the shape a little bit as well. But yes, the channel is very small. And then the size itself, I can actually show you how big is the model. So yes, so this is one of the models we were using. Yeah. Okay, maybe I can do it like this. And then the spiral shape is the cochlea. So then the entry is very small.
So it's about the size of a pea, we like to say. So it's a very small kind of tiny structure. So there's a lot of work in also thinking how you about the design as well. Because once you've got the cochlea, it's in a kind of a strange orientation, or it's in its natural orientation. But in terms of understanding what the forces in different relevant directions, we'd have to correct for that within our kind of processing pipeline. So we built a whole pipeline within MATLAB where we would take all of these micro CT scans, segment them with this template-assisted segmentation, then go into a MATLAB implementation of slicing the cochlea, understanding the cross-sectional area, making equation effectively for this like characteristic spiral, and then correcting that and making it directly into a print. So we could get at the end a STL file we can directly send to the printer. So then we can have everything very consistent and ensure we're like accommodating for things in the right way, because depending on how you slice things, for example, we'd have to take away the little membrane you'd usually insert the cochlear implant, because it has at a kind of oblique angle, where that would also play a role where if we want to look at just the shape of the cochlea, we want to isolate all these different variables and increase the complexity as we'd go, effectively. One of the key pieces of post-processing we were looking at also was improving the transparency of the models. So then we could understand where the positioning of the implant would be and relate that to the force as well. So otherwise, it's hard to know. I mean, within the actual normal anatomy or cadaveric specimens, you're putting it into a small bony channel, and you have no idea what's happening inside. So the beauty of a 3D printed model is that you can actually see what's happening to the implant. Is it bending over on itself? Is it buckling? And try to relate that to any features that you see in the force, but also in the kind of electrical modeling as well.
And then you also mentioned that you had to do some sort of coating so that the internal surface can be closely mimics the internal surface of the skin of the inner ear. How did you guys accomplish that with your printed models?
So we first out tried a couple of different coating techniques. So we used, soap, a solution of soaps. We tried medical oil, like silicon medical oil. We tried lots of different things. And then we tested out pluronic, which is it polymer based, right? Iwan, how is it called?
Yeah, it's a PEG-based surfactants, effectively,
PEG-based solution, which we had really good results with, and it helped out to create a very thin, nice layer, which then resulted with combination with a solution in a very similar insertion force profile as in cadavers.
Did you test other fabrication methods besides 3D printing?
We were interested in casting. There was a time where we were interested in casting, and there were papers discussing the casting. But what I quickly disliked about that is that it just takes a lot of time, and then the variability. I mean, the problem there is that you a cast material within the bone, like the temporal bone, into the cochlear channel. And then you dissolve the bone itself, and then you take that part, and then you cast around that part again. And so it's a bit more hands-on rather than direct 3D printing, and that also introduced a lot of variability. So the problem is that then creating the same model 10 times could create deviation because there is lots of manual work. So that's one thing which we were a little bit worried about, the post-processing, or specifically the coding, because the coding was done manually. But then we did lots of nominal actual analysis using micro CT again to rescan what we already printed with the coating in and compare it to the original CAD model to see what is the actual deviation. And we were pleased to see that the deviation was minimal for our use case.
Yeah, and I guess the issue with that the casting techniques is this is destructive, right? So once you make that one model, then that's it. Where with the beauty of 3D printing is once you have the file, you can manipulate it. You can add different channels for flushing water or whatever you need as many times as you want, so you can replicate that file. And using that nominal actual analysis Filip showed, we could get down to about 30 microns of deviations of the surface, which is the resolution of the printer, the CADWorks printer. So we were very happy with that, really, that we could get down to that level of resolution or accuracy of the surface, I should say. Yeah, and in terms of other techniques we also looked at, we looked at stereolithography, like classical stereolithography techniques, more to make a Cochlea-on-a-Chip model. So this is more of a microfluidic version of the cochlea that we've also worked with, where we'd want to make a flat cochlea where we could inject cells, have them inhabit the model as they would in the real anatomy, and then look at how they electrically behave when you stimulate with Cochlear Implant. But the issue with that was, although it could probably get you smaller, very fine features, you couldn't get the size of like a 1 millimeter high channel, because that's very difficult to make with those. So it's kind of strange, actually, for this scale. It's a hard scale to work with because it's not small enough for a lot of microfabrication kind of techniques, but it's not big enough for a lot of kind of standard 3D printers or other kind of fabrication techniques. So it's kind of this middle scale, which is very hard to engineer with, but it's an interesting challenge.
We also tested out the SLA printers. I think, and to a certain degree, it worked, but funny enough, with the CADWorks one, we were able to achieve, like, even though the resolution on paper was higher or lower, I mean, comparing, let's say, 20 to 30 microns, we are still able to achieve better overall deviation from the model. So the deviation over 90% of the surface of the model was actually lower with the CareWorks printer compared to other SLA printers. And we were also studying why is this happening, and like how is it possible that DLP achieves better resolution. So yeah, it was a lot of interesting things, and I think that one part of it is also the material itself. So we're using the microfluidic one, the CareWorks microfluidic, and what we like there is that the viscosity of the resin is actually lower, so it's easily flush out from the channels as they're being printed. Because what we found out is that if the viscosity of the material itself is higher, then the resin is trapped inside during the printing, and then when you create another layer and another layer, let's say in a channel, then as the resin is trapped inside, the curing actually penetrates also a little bit inside of the channel, which creates the deviations. So yeah, that was one of the things we discovered, and we're trying to publish now.
I think there are two things that Iwan had said that were really piqued my interest. One was the fact that you guys had attempted to sort of do the modeling on a stereolithography. Like where I understand that you weren't able to get the size the scale that you wanted to work at, but then do you know, like, would you have been able to duplicate the work that you did on the 3D model? Like, what part of the work would have been translated onto that platform? What was it that you were trying to do on that one?
Yeah, so I can maybe explain a bit more about the Cochlea-on-a-Chip that we were looking to make for that. So this would be effectively we needed a PDMS microfluidics chip that we could put then on a glass substrate, ideally a micro electrode array, which is effectively a bunch of electrodes which are patterned on a glass substrate, which can then connect to an MEA rig, which can record from all of those little electrodes and record the action potentials from individual auditory neurons in our case. Because actually embedding a lot of the electrodes within and culturing cells within the 3D prints is quite difficult. And if you can use glass substrates where everything is already well established, then it makes it much easier, because you need every chance you can get with these types of cells, because the extraction of the cells itself from these primary auditory neurons is very delicate, and a lot of the time you need to be able to handle them correctly and culture them into the correct conditions. So we were able to get a slightly simplified version of the structure of the cochlea within a microfluidic chip, and actually embed small channels that replicate some of the porosity of bone that we have in the real cochlea, between that channel and a channel which can contain the cells. And with putting a real cochlear implant inside of this microfluidic chip, we could see how the cells would react to it, so how they would electrically respond. And it turns out that we actually focused a bit more on patch clamp electrophysiology, which effectively is effectively stabbing a single neuron with a small needle and delivering a very defined amount of current or different current patterns, which other members of our lab, so it comes with Gillies, she did a lot of the work in terms of extracting the cells and culturing those, and Paul Charlesworth, who worked a lot on a wealth of experience in patch clamp electrophysiology, and he comes more from a kind of bit of a bioengineering background, as well as someone, Sarantos, who did a lot of work on purely kind of computational work and lots of more engineering work, but could make all of these different paradigms of different electrical pulses effectively. So we can really, there's really good collaboration between us to see how you could deliver all these different types of patterns in a really high throughput format, to understand how the cells would react to these different electrical impulses, see how you can make that interface as efficient as possible. So yeah, it was a really interesting kind of multidisciplinary project, and interleaved with some of the more larger more accurate cochlear models that we've had, which we introduced pores into the full cochlear shape to mimic the electrical properties of the cochlea. So we designed different pore structures directly within MATLAB again, to basically have voids within the model, and by filling that with saline, which is conductive, and the resin plastic, which is non-conductive, then we can tune the electrical conductivities of different parts of the structure around the cochlea and mimic the
basically by tuning the size of the channels, we could change the current spread within the model. So one part of the research, what is interesting is to see how the implant stimulation spreads through the spiral shape, because as you have the implant inside, and then it has 16 to let's say 22 electrode plates, each plate is being stimulated at a certain frequency. So basically you stimulate the plate, and that stimulates a certain amount, or certain degree of neurons, which then transfer it into a specific frequency. So this way, what we were studying is that if you put the implant in and you let's say stimulate electrode number 13, how does the current spread, and does it also affect different neurons that it should in a certain range?
And so where's that research right now? I guess, has there been a publication on that, or is that ongoing right now?
It's so Chloe finished her PhD I think this year or last year, ending of last year, and this is being published as we speak about it.
Oh, great. Amazing. So the other question I had was that considering that you've been able to do such amazing work over the many years using 3D printing, there must have been some significant gaps, or at least minor gaps, in the use of 3D printing that you would have felt that, if we had this in place, it would have been either better, or the data set would be a lot more clear without any of the any outliers or anything like that. How would you guys address that?
Mm-hmm. I think that one of the key drawbacks, at least in my research, was that the cochlea, or the spiral shape like object, the scala tympani itself, it has this very thin layer of soft tissue inside, which changes the insertion ever so slightly. And what would be amazing to have a model where you could replicate that soft tissue inside of the model. But at the moment, we might try to replicate this with multi-jet printing technology. But the problem is that the channels are so small is that it will probably result in significantly higher deviations. So that was one thing. So looking at the multi-material properties, following up on that, being able to print conductive material, that would be very interesting for lots of reasons, specifically with the research about cochlear implant stimulation, looking at where the current spreads during the stimulation. That would be interesting.
Because, so to pick up on that point specifically, to look at the kind of electrical spread we've actually focused a lot on the kind of designing small channels to just manually feed wires into the models, and then doing post scans to understand like exactly where those wires are. But if you could make electrical tracks within a 3D structure that small, then it would be it's a significant challenge. So it's completely understandable why it's not really possible with any technology, even multi-jet technologies now.
I think it's amazing. I think on our side, we always find the hardware side is one that has a very defined trajectory. Meaning, in terms of scale, you're always pushing scale to its limit. The material side is almost a blend of chemistry and being a fast order chef at a fast food restaurant, because it's people. I think the user base, especially amongst biotech and the larger biology base, I think the need for a wide range of very unique materials is immense. Like pretty much anything that conductive material, I think, has become one of the most it's sought after material. But what you're asking for is not broad spread conductivity. You're asking for being able to embed a thin line or a conductive material within a larger matrix, which it'd be nice to have something like that.
Yeah, but even just having good flexible materials that replicate the kind of small membranes within the cochlea is like a very important question that we've really struggled to fabricate. And a lot of this stuff you can actually mitigate with design considerations. So we've looked at effectively sandwiching different membranes between different prints, or like having smaller channels that you can feed wires in, for example, or feed conductive material in, or even for example, for some like adding colors to make like a nerve, so you can identify where the nerve would be within a clear model. We found ways to kind of get around the kind of limitations, and a lot of that has just been in the geometrical design. So even in the pore structures I mentioned before, really we've made it so, okay, you've got this non-conductive material, but by the way that you construct the actual layout of the material, you can actually play with electrical properties. So there's always, most of the time you can find ways to get around things just with some creative thinking.
When did 3D printing actually become prominent in cochlear research? I assume it's more of like an emerging technology, or has it been kind of sprinkled here about previously?
Yes. So in the literature, like a couple of years ago, they started experimenting 3D printing the models, testing different technologies. But when I checked, the problem is that they didn't really check what's the deviation from the real model, so from cadaver. So then it's difficult to see like if that technology worked well or if it didn't work well. So, but yeah, the 3D printing started a couple of years back, like basically with the first SLA printers. So let's say, I think it was like around 2015 of things like that. But then creating a model which is fully characterized, and so everyone else can print it, and then seeing what's the deviation between, like using the nominal actual analysis as a proof that's fairly unique. It's been done for different use cases, but as far as we know, not for cochlea.
What were the models look like before 3D printing? Was it like primarily physical? Was it like animal?
Yeah, exactly as what you just said. So it was cadavers, so cadaveric temporal bones. That was one thing. The problem there is that there is a difference between fresh cadaver, fresh frozen, or
Oh, fixed cadavers as well.
Fixed cadavers. So there were these models. Then there were models which were casted. So as we discussed before, there was the technique there, and there were also milling. So one of the techniques they were using is instead of creating a 3D model, they created either a 2D model where they cast, let's say they have a 2D spiral, and then they cast acrylic around it, and then just pull out the material itself, which create a spiral, but it wasn't rising spiral. It's just 2D spiral, or anything else. That might be. It's like, as Iwan already touched on this. Like the problem with cochlea is that it's too big for microfabrication, but too small for normal type of 3D printing. We were also checking out FDM printing. We were checking out SLA. We also checked two-photon polymerization, which is a very high-end microfabrication technique. And I mean, again, the problem is that the cochlea itself is too big for that type of technology. So you can create, like nano lenses and things like that, but it's exactly the range where you need to have something which can create something, let's say, in centimeters size, but on the resolution of below, like, let's say, 50 microns. So it's it's a challenging one.
Out of curiosity, did you both have experience with 3D printing before you started working on cochlear implants?
I had about a year with the RSO jet printer, which was very specific, and during that year I was also testing out the SLA printer, but not to not too much.
Not extensive. Yeah. How about you, Iwan?
Yeah, so I'd collaborated quite closely with a friend who was doing a lot of bioprinting at the time. So he was using more extrusion-based printing for polymers, as well as for gels embedded with cells. So I helped out a little bit in terms of imaging those, like playing around effectively. And we also at the time had a kind of startup idea, which was mainly for competitions and learning, where we'd look to make a 3D printed nerve-based conduit, which would help the bridging of nerves from nerves that suffered from traumatic injury to regrow effectively. So I was looking at more of the gels and the biomaterials aspect. He was looking at the 3D printing aspects, and other people were looking at the neuron kind of outgrowth as well. So it was a quite interesting project. But so that kind of sparked my interest into 3D printing, and actually when this opportunity came along, as well as my interest in kind of neurotechnology, kind of is a good combination of those interests, really.
I always found, interesting that 3D printing allows you to, create something in CAD and then just have it physically in your hand. And it was one thing I really like about my PhD is it was applied research. So we designed something, the next day or in a couple of hours we had it physically, and we could see what it actually is and how it works. Because it's also difficult to appreciate how things work specifically in inner ear and how small they are. Because if you work in CAD, for hours, you've got everything 200 times scale up, and
just zoom in and
everything is huge, and you're easy peasy, blah blah blah. And then once you 3D print and see it one to one, then you actually appreciate how delicate the structures are and how amazing is the anatomy of human ear.
Yeah, and that's one thing we found with the anatomical models that we've been making for more education purposes, is that there's been a huge growth in terms of virtual models, VR, AR kind of models to understand kind of the anatomy, which especially for like surgeons is really important to know the visual spatial kind of way, how they relate to each other, or the size and shape. But there's nothing quite like having something in your hand that you can actually turn around and visually appreciate and really see the scale, because as Filip says, like, you can zoom in as much as you want on a virtual model, but you sometimes kind of can miss the scale. I know, like, a lot of the times I'll make something in CAD, but actually it's like, oh, it's this small, rather than like I think about this big. Yeah.
Yes, you do get tunnel visioned, and you get the blinders go on, and you forget scale at that time, and until you print it out, do you realize that really I thought it was a lot bigger than this, and I can't see it, or I just broke it, and that usually is either one of the two.
Yeah.
So, and now that you've got all of this work behind you, and you have become you brought solutions to the table. What are the next steps in terms of the current methodology that you've developed in order to evaluate cochlear implants? What's the next step for you guys in terms of research? And then, give us some idea of what the next steps are in front of you.
Yeah. Go ahead.
Yeah. So I guess from the research perspective, we're really looking at so we focused a lot on having individual cochlea that we manipulate the shape of, and now we're looking at really expanding that into all the different shapes and sizes that we can get of a cochlea from that kind of database that we've segmented, as well as looking at other factors for like the surgical considerations. So looking at the surgical drilling site and how you can actually effectively insert the cochlear implant as well. And then we're also still looking at trying to improve the Cochlea-on-a-Chip, microfluidic chip version I mentioned, and really take a lot of those learnings and combine them with some of the computational models that we have, so you can understand from each different type of model, you have its limitations, be it computational, be it physical, or even the cadaveric kind of real tissue. It's usually a trade-off of the amount of detail you can interrogate and actually measure the model versus the accuracy of the model. So validation is kind of key. And on our other side, like we've started a small company, COSA Ltd, that really spun out from a lot of the expertise that we generated from this, and we collaborate with some of the cochlear implant companies, different clinicians, etc. Through our work, and through those connections, we kind of we developed these different projects together, and we're actually getting some traction on that side to look at all sorts of different things in both the realm of the ear, or mostly a realm of ears, be it more educational visual models, or R&D projects to look in detail how like the pressures, forces, and things vary for medical device development and drug delivery, for example. But then we're also looking at making really realistic phantoms more generally as well, for head and neck to start with, and a little bit of everything. We've even printed a mouse for a local startup who were developing different devices to look at mouse tumors. So a little bit of everything.
So now, I guess there are two things I wanted to address. The first was the current work that you've done and the procedures and the papers that you've put out. Do you see this being applied to other applications? Like there's cochlear implants. Are there any other implants, or are there any other surgical procedures that you can see that this type of procedure could make it more successful, and provide a unique research point for others as well?
I think that what is at the moment rising quite a bit is the drug delivery. and treating the cochlea or the hearing with drugs instead of cochlear implants. So cochlear implant is sort of like a universal solution as far as you have auditory nerves in cochlea functioning and you've got neurons there. You can most likely stimulate them using the cochlear implant. But the problem with cochlear implant is that, first of all, it can create, it can damage the residual hearing. And second of all, it's permanent. So it'll be there for forever. So some diseases and some hearing loss can be treated with injection of, for instance, a protein. So there is a delivery of a protein which is specifically connects, for instance, neural pathways. And by just introducing this protein into a cochlea, into certain regions of the cochlea, the patient can hear again without the need of cochlear implant and having a device inside. So now there are companies, pharmaceutical companies, looking at these drug delivery systems, and that also uses an insertion, because you need to either insert the drug into the middle ear and then through the tympanic membrane, through the round window membrane, the drug gets inside of the cochlea, or you can insert the catheter into the cochlea itself through the round window, as you do with the cochlear implant, and inject the drug this way. Each technique has its advantages and disadvantages, but this is how you can leverage our research, let's say, in the insertion forces to optimize the insertion of the catheter into the cochlea.
Yeah, and actually, just to add that the professor that we work with, Professor Manohar Bans, he has the claim to fame of being one of the first surgeons to perform gene therapy in the ear, which is really exciting. And actually, there've been quite a few news features about it to show the first patients who actually have like really great results coming out from that. So these gene therapies have the potential to be curative. So especially for very specific genetic hearing loss, then they're really exciting in the way that things are going.
And the research that you guys have done in terms of the insertion forces would be the driving factor. Am I correct? Or that would be something that they would be able to use in order to apply those therapeutics?
Yeah, so there you really need to be very careful of maintaining the cell population, right? So you really want to reduce any trauma that you have during the delivery of any like a drug or gene therapy or even cell therapies. There's quite a few companies in that space as well. So making sure that you don't damage the residual tissue is really key to ensure that the therapy is successful. And there's a lot of work both on the not just the kind of surgical forces itself, it's understanding of the anatomy and what are the key features that you kind of extract and the surrounding anatomy as well. So even making kind of surgical training tools to allow the training of these kind of new novel kind of surgical techniques is very important, I think, for us. And
amazing.
I think.
And then the second part is this new company that you've started. Why don't you tell us what is the mission statement of this company? What are you guys hoping to achieve? What is the current research or the direction that you are hoping to move ahead in?
Mm-hmm. I think that we started this company based on our research and based on the interest of medical device companies in our studies. So basically how this all started, we mainly create anatomical models and surgical training tools for ENTs and specifically in general for head and neck. So we started with ENTs and with the inner ear anatomy because that's where we have most experience. But we are also working on other projects, including the head itself, or neck systems, or nose and models like this, where you've got like.
Oh, wow.
So we're trying to also differentiate and not being only in the ENT realm. A couple of exciting projects we are working on right now, and we think would be really nice. For instance, drillable temporal bones. So we identified that there might be a need for these drillable temporal bones, as ENT surgeons typically need to undergo a couple of these courses and try to drill the temporal bone, create mastoidectomy, and see how they can access the inner ear. The problem with this is that these courses are run only a couple times a year, or over UK and like globally as well, and they use cadavers. And the problem with cadavers is that they are scarce and expensive, and you need a certified lab to run cadaveric courses. And so what we're trying to achieve with these models is to create a cheaper alternative for the drilling. So then, let's say junior surgeons can practice more on these bones, which we optimize to behave as the real bone during the drilling. So the feel should be the same. The bones are again coming from micro CT scans. So the interesting thing about the temporal bones is that it's fairly porous. There are lots of air cells inside. So when you're drilling through, the feel is different than if you drill just through solid plastic. So there was a challenge to replicate these. And we are still optimizing these models. And what we're trying to achieve with this product is to really create a validated model, which is sort of certified by the ENTs to behave and to be a good replica of the cadaveric models. And again, the advantage of 3D printing is that we can introduce malformations. We can introduce unique cases into each model. So then every participant of the course can practice these unique cases, which can happen in 10,000 patients, so very unique cases. Because the problem with cadaver is that, you drill it once and that's it. Yeah. And every cadaver is slightly different. So what you can achieve with this is that everyone can start with the same, everyone can start with this malformation or that malformation, and then you can move to cadavers. We don't want to be, a competitor to cadaver, because cadaver is still the real thing, right? But having something where you can practice a bit more, we think could be really, really good.
So the models you made, were you the ones designing it? You printed it, I assume, and then you approached the EMTs or the institutes yourself? Or was it kind of the other way around, where they approached you with very specific designs and needs, and then you fulfill those?
Yes. So as we were working on one of the other projects for medical device companies, one of the directors of that device company asked us, if we happen to have these type of models, and we started to question, if there is a market, if there is a need. We talked to our friends and colleagues here in Cambridge, because there is a fairly decent ENT community. So we were talking with them, seeing what could work or what couldn't work, and we started prototyping. We started prototyping to see if it's even manageable to have a plastic or 3D printed model to behave as a bone. And currently we are at the iteration 44 or something like that, and there is always always space for improvement. So basically what we're trying to do right now is that we were part of a cohort in Birmingham, where we tested out the bones with the ENTs there. We tested it twice here in Cambridge. We are also running tests in Czechia, in Prague. So we're trying to get as much feedback regarding these bones and optimize the products, and see where is the market, and how we can make it the best possible product, basically based on the feedback from real surgeons. And technicians.
Yeah.
So it would seem like as if this bone material that you have could be used pretty much in a wide range of applications. It could be used for the temporal. It could be used for any. You can change the density of the porosity of the bone material to mimic pretty much anything. So it could be used by any surgeon or any doctor throughout their studies.
Yes, yeah. I think that we also started with one of the hardest tasks because the temporal bone is so unique and the anatomical structures are so small. So it's I think we started with the hardest one, because printing a femur or other bones which are less difficult would be easier. But where would be fun in that?
No. There would be no fun in that. You're right. So now, I guess, can you? I think through your journey, you have probably learned a significant amount about yourself, about how to work in collaboration, in terms of identifying goals, and then sort of understanding that the issues that you run into, whether they be problems in order to procure funding, or to get adequate the right tools, or anything. What kind of things have you guys learned throughout this entire process?
You want to do
I mean, I guess some of the lessons that I really kind of appreciated are the importance of validation and the importance of getting lots of different approaches to the same problem effectively, because no one model will solve every problem. So being able to use let's say cadaveric models or computational models and use kind of the same analog in different ways has been really important, and actually getting the validation, as Filip was saying, and doing that independently to show okay, this is actually really replicating the same thing. Because it's very easy to make a model, but how do you actually quantify it? It's true to the real anatomy, that you can trust the decisions that you're making from that model. Because there's always limitations. And understanding those kind of limitations is really key. And there's also being a little bit creative with the design aspect, I think is super important. So understanding of okay, well, this is the limitation of this technology versus that technology, or how can we split this model into different component parts, have a little bit of more manual steps if needed, or go into different kind of multi-material kind of printers or different kind of things when we need to. Because there's also considerations even with, let's say, the drillable models that Filip just mentioned, of cost. So it's not just making the best model, but especially for clinical schools, which have where you need to have this as a consumable item, you need to think about how do you make it a cost effective and at a reproducible level, where potentially you'd need kind of tens or even hundreds down the line of these models. So how do you make it? You can make one very very nice model, and the most effort is going into making that one model, but then you also need to think about how do you make that reproducible, and even just making the post-processing very easy to do, easy to perform, and efficiently for the models as well.
So do you provide that entire workflow, from the design, teaching them how to do the 3D printing side, the post-processing? Is that what part of Costa does?
Not really. So what we do is we create the final product. So we have basically two branches. One is doing the consulting work. So how that works, that usually the medical device company or any company approach us and ask us if we can create something, and then we put our heads together and we try to think about how this could be done and if it's actually possible. That's one way we work. The other way we work is that on the background on creating these bespoke models for companies, basically the consulting work, we create our own products, and we do our R&D on things which we think could be interesting and could have a market. So then basically in the evenings we started running, lots of experiments. And I wanted to follow up on what Hemdeep asked about any recommendations. I think like just a practical example is that, start with something small and very easy, very, like not complicated structures, and create it at least 3 times, and then measure 3 times each and go this way. Because what happened many times, especially at the beginning when we were starting with 3D printing, we sort of went too much in the future. We created like this incredible difficult model. We printed it. It worked perfectly, but we were unable to replicate it. Every time when we tried to print it again or replicate the same thing again, it was basically impossible. Like we like there was always a problem. And then going backwards, it's significantly more difficult than going forward from something small. So I think it's like a very casual sort of thought that you should work with a small thing and build up on that. But like in our case, like that's 100% true. So yeah.
Maybe one more thing to add really on the kind of, which goes into kind of an approach we've also now taken with Costa, is understanding like the ecosystem as a whole, because as well as working with medical device companies, we work more directly with clinicians as well as like, let's say, medical schools, and all of those bring different different perspectives, but also different value in terms of understanding how you validate the models directly with clinicians, and you have different ideas coming from those, and then also understanding practical considerations going through with medical device companies and getting access to really high quality scans or like materials that we can then scan through medical schools that then can be used, made more available, because these things are like hard to come by. You don't get cadaveric specimens of ears everywhere, right? So being able to democratize access to these kind of models is really important to us. And we've even had lots of kind of serendipity along the way, like just seeing with a pediatrician, saw one of our ear models at a conference, and happened to be, it was actually not in Cambridge, they happened to also be around Cambridge. And they had a very interesting technology looking at making effectively a microphone which would feed into some bone conduction headphones for kids with recurrent ear infections. And someone actually had started calling their microphone, one of the 9-year-old girls actually, called the microphone their extendable ear, because it's something from Harry Potter, that one of the Weasley twins have an ear on a string, basically. So they asked, can you make this make our microphone into an extendable ear? So we designed like the ear to be like a sleeve around the microphone, and then she conducted a whole study with the kids to see how they react and see like how they engage with the technology. So there's a lot of really fun things which actually like helps. Like, even though it seems kind of silly, it's actually really informative to help kids to engage with it. And she got another grant actually to then take those ears to Malawi as well. So she does a lot of work there. So there's now about 30 of our ears that are now in Malawi being used with these microphones, which is quite cool. So it's a very sweet story, and seeing the photos from it was amazing. Where especially like there's small considerations, like they wanted to have them pierced. So then there were a deaf community in Malawi that were making earrings. So they can put the earrings on the models with the little microphones. Yeah, so it's a really touching story.
So you guys have had a very interesting 5 years. It seems that it went from not having really much idea of exactly what cochlear implants are about to now running a company that is providing significant and interesting solutions that is cutting edge, and you're reaching out to an extremely wide base of users and patients, and it's an amazing story to hear. I think one of the mission statements that we have as part of our at the office is the fact that, we see 3D printing as this ability for people to researchers and users to accelerate, the idea, to iterate, to design, to retest, and so on and so forth. But eventually all arrows lead to one thing, and that is to reach out to a larger audience, either through a commercial venture, through engaging with new techniques. You guys, it looks like as if you have done it right, and you've been very successful. I really do congratulate you for that.
Thank you. Thank
you. No, thanks for that. Yeah, and I think actually that comes back to like all the way when we started kind of looking at that 3D printer that we mentioned. We actually just got the Cochlear kind of models and sent it to a bunch of different companies and seeing like, okay, who can print it the best, basically. So, and then from there, it's just getting the hits in, right? Like doing so many different iterations that you learn on each failure and each success.
Yeah, for sure.
And then you just improve. If you can improve like 1% each time, then you can really improve like your outcomes down the line.
Well, this was a fantastic inaugural podcast. I'm so happy that we had a chance to talk. I think it's everything that I thought it was going to be that first time that I met you at Cambridge. And I think when you had presented to me exactly what you were doing, it was amazing then, and now to hear what you've done since, it's fantastic to hear. And so I do thank you for taking the time out today to sit down with us. I think it was extremely meaningful for me and to Robin, and I know our team is very appreciative, and I'm sure that the audience that will hear this will also appreciate the work that you guys have done.
Thank you. Thank you for having us.
Good. No, and also thanks because it's been great to have the support of CADWorks, because it's one of the first printers we really had like hands-on experience with at the start. So it's like really kind of the start of this journey, which has gone in a lot of different wild directions as well. So it's been really good.